Should we get started, Pascale?
Sure.
Okay. Well, welcome to the QBI Online Seminar Series. I'm Judy Sakanari, who will be introducing today's speaker, who is Dr. Pascale Guiton, who is an assistant professor at Cal State University in East Bay, actually just across the East Bay, if you can see through all this dark smoke. Before Pascale gets started on her talk, I'd like to just give you a brief bio sketch on Pascale, who is truly a dedicated and passionate teacher, mentor, and scientist. Pascale did her undergraduate work at Georgia State University in Atlanta, and she actually got a very early start on her research because she started working with Streptococcus pyogenes and the molecular mechanisms of iron acquisition by this bacterium. And then she went on to graduate school at Washington University at St. Louis, and she continued her work on the molecular microbiology and molecular pathogenesis of other bacterial species. But then she saw the light. She wanted to study parasites, and she joined John Boothroyd's lab at Stanford University as a postdoctoral scholar, where she has been working on the protozoan parasite Toxoplasma Gondii. And she's been working on toxo since then, and she's going to be talking to us about how toxo infects new host cells. And I'll turn it over to Pascale. And just while she's getting ready to share her screen, just want to mention that we'll have the Q&A at the end of Pascale's talk. Okay.
And present. And I'm going to put the caption on, just, you know, my accent and all. Caption and pointer. Great. Well, thank you, Judy, for this invitation and this introduction. I'm really super excited. You cannot see it, but I am. Usually, I'm the type that feed off of my audience, so I have you guys in my head, and I will be clapping. I'm just joking. So today, I'll tell you about this question that is really the driving question of my lab. How does Toxoplasma initiate infection in a new host? And the answer is many mechanisms. How they do it, we still don't know. So at the end of this talk, you're not gonna get the answer. But we're gonna tell you, I'm going to tell you some of the approaches we are using to try to get at this question. And on this immunofluorescence image, what you're looking at is Toxoplasma in different stages. So the green is a stage that is called the bradyzoite, and the red is a stage that is called the tachyzoite. And we're gonna talk a lot about these two stages throughout the talk, and I'll come back to it. But I love it because it's one of my first toxo images. So when I graduated from bacteria, from prokaryote to eukaryote. Anyway, so toxo is famous, okay? It's like the king of parasite as far as I'm concerned. And, you know, it's like the Kardashian of the parasite. So it's been talked about in the New York Times, CBS, The Atlantic, and so on. And a lot of you have heard about the cat parasites that mind control and, you know, make the cat lady crazy. Well, there's some evidence about the correlation that the parasite causes mental illness and schizophrenia in human is quite, it's weak at best, okay? But in mice, we do know that Toxoplasma infection does alter the behavior of the cat, of the animal. But it's one of the most successful infectious agents with a known prevalence of over 30% in the world. And in this audience, I think that when I last checked, it was about 34 people. So 10% of you may have Toxoplasma in your brain and you don't even know it because the parasite just is asymptomatic for the most part, unless you become immunocompromised. And so here it is in all its glory. It's an obligate intracellular parasite, And right now you're looking at an electron microscope of the parasite inside a cell, inside a parasitophorous vacuole. So this white space is essentially the house where the parasite lives. And its name, Toxoplasma, comes from the Greek toxon, which means bow, and plasma, which means form. So it's essentially a bow-shaped parasite, okay? And with my neighbor, I call it the banana parasite. And again, it's a single-celled obligate intracellular parasite. And it was first isolated from this cute rodent called the Gondii in Morocco. And what is really fascinating about this parasite is this apical complex, right? It is critical for the parasite invasion into the host. And the reason is that it has those specialized organelles. So you have those long one that looks like baseball bat. They're called the rope trees. Then you have those, they look on this side over here, they look like cigars, and they're called micronemes. And then you have those dense vesicule, right? Those dense shape that are dense granules. And so all these are involved in the parasite ability to invade the host cell, to evade the immune response of the host, as well as modulating the transcriptional profile of the host. And what is cool is that Toxoplasma belong to this phylum Apicomplexa, and all of these parasites are essentially characterized by the presence of this apical complex. And in the family, in the Apicomplexa phylum, you have Plasmodium falciparum, right, the causative agent of malaria, and many others. And all of the parasites in this phylum cause disease, okay? So they are of veterinary importance and medical importance. So for instance, Eimeria affects chicken, and Neospora, which is not depicted here, affect dogs, and then of course Plasmodium. So Toxo exists in different developmental form, okay? And the three infectious stages that are capable of infection in humans are the sporozoites, the tachyzoites, and the bradyzoite. And all of these forms have these, this apical end, okay? But we know a lot about the content of those proteins, of those virulence factor, usually, in the tachyzoid, but we don't know much about it in the bradyzoid. And that's because it is very difficult to study the parasite in this form. It's only until this year that we were able to develop an in vitro system that can recapitulate the sexual cycle of Toxoplasma in the lab. And that was work done by the Knoll lab. But anyway, so these are the infectious form of Toxoplasma. But how do you get infected? Well, it's pretty, Toxo is pretty wild, all right? So the definitive host, which is the host in which Toxoplasma undergoes its sexual cycle, are the feline. And this includes the cats and the big cats, right? So domestic cats, big cats like lion, tigers, whatnot. And when they get infected, they shed in their feces unsporulated oocysts, right? So you can think about these as like the spores of the parasite, if you will. So this in the middle here that I'm showing it is the form that gets shed in the environment. So they are unsporulated, and this blurry structure inside is essentially contain the zygote, okay? And in the environment, there are processes that we do not understand. Well, actually, this one is the unsporulated oocyst, and then this is beginning to sporulate. Anyway, in the environment, there are processes that we do not quite understand. The oocyst will sporulate and produce 8 sporozoites, okay? And there's 4 sporozoites per sporocyst, right? And then these oocysts would get ingested by, let's say, a rodent, a rat, mouse, and then it will, in these intermediate hosts, the parasite will get released and undergo its sexual cycle. And the animal will get sick, and eventually the parasite will change form again and establish itself in the brain and muscle tissue. And you will get the formation of these tissue cysts, this is one that was isolated from the brain of a cat, actually. And then via predation, the parasite goes back into the cat. So this is essentially the life cycle of Toxoplasma, but clearly we care about it because it affects us as well. So you can virtually all warm-blooded animal can get infected with Toxoplasma, either by ingesting oocysts or by eating undercooked meat that originate from chronically infected animals. Okay, and in California, at some point there was the sea otters that were dying, and Toxoplasma was actually the cause of that of the death of the sea otter, and that was work done by in the lab of Conrad lab at UC Davis. Anyways, so what happened after you have ingested either the oocyst, right, because you just didn't wash your hand and you had a cat that was infected, or you like your steak rare, but it came from a cow that was infected with Toxoplasma. So now we're going to go inside. So you've eaten either the sporulated oocyst or the bradyzoite. So in the GI tract, the parasite will basically get released via the actions of pepsin and other digestive enzyme, and will infect enterocytes, so intestinal epithelial cells. And then very rapidly, the sporozoite will convert to tachyzoite, and the same is true for the bradyzoite as well. They'll convert to the tachyzoite, and so tachy means fast, right? So you can think about tachycardia. So this form of the parasite will divide rapidly and lyse the host cell and goes on and infect neighboring cells and disseminate throughout the body of the host, okay? Now, via mechanism that we don't quite understand, but we know to involve the immune system, the tachyzoite will switch back to the bradyzoite stage and remain in the brain, as I said, and the muscle tissue of the infected individual. And so unless you as a human get eaten by a lion on a safari, chances are you will not transmit Toxoplasma. And but if a mouse gets eaten, it's just the whole cycle happens again in the animal. And so I want to point out that the tachyzoite form over here is the form that gets transmitted from mother to fetus, okay, during pregnancy. And it's, we can talk more about this vertical transmission, but that's the form that is dangerous for the fetus. And so we know a lot about tachyzoite interaction with the host cell. We know a lot about it in the mice. We know a lot about it in vitro, and we don't know much about this side. As a matter of fact, we don't know any of the molecular players that are involved in this first stage, okay, of the infection. And so what my lab focuses on is trying to identify molecular players that are involved in this very first interaction of Toxoplasma and its host, and how the parasite is able to differentiate so efficiently from one form to another. And so the way I divided the talk is, first we're gonna look at the work we've done in trying to identify factors that are associated with host-parasite interaction, and then a short story about work we're doing looking at differentiation, but from a metabolic approach. So I told you that we don't really know much about how Toxoplasma interact with intestinal epithelial cells, at least from the sporozoite and bradyzoite form. And so what we've done in collaboration with Fritz's lab at the University of Washington, we basically infected mice with the tachyzoite form, allowed the mouse to become chronically infected, and then took the tissue cyst and fed that to the two kittens, and then allowed the kitten to shed oocysts. And then we took these oocysts and removed the allow them to sporulate and extracted the sporozoites from those oocysts, okay? And then we use this fresh sporozoite to perform a transcriptomic study in intestinal epithelial cells. And so just briefly to go over this, we had rat intestinal epithelial cells, IC18. They are non-cancerous, so they are derived from the ileum of newborn. And so we have the freshly existed sporozoite, we have sporozoite that we have killed, and then we have tachyzoite control. And so we infected the cell for 8 hours, and we chose 8 hours because it is right before, or it is well before the sporozoite switched to tachyzoite, and we verified that by immunofluorescence microscopy. And then we basically generated RNA and perform RNA sequencing on it. And so we published this, so I'm just gonna give you a summary. So what we found is that sporozoite infected cells actually have an immune signature. So it's an NF-kappa B-like signature. And this was striking because it was hypothesized that the sporozoite, because they don't divide and they're shortly lived, they probably don't induce an immune response. And so it was quite interesting to find that the immune response that we know well following infection with tachyzoite actually is initiated very early in infection. So what are the factors that are associated with inducing this immune response or with regulating this immune response is not known. And so this is where my lab, essentially this is where my lab starts, right? So what are the effector proteins that are involved in this host pathogen interaction, the interaction of Toxoplasma with intestinal epithelial cells? And I told you that Toxo has this apical side that have all these organelles. In the tachyzoite, we know that the content of those organelles are required for virulence, okay? So they're involved in attachment, in invasion, in modulating host immune response, in egress. And so because sporozoite and bradyzoite have these organelles as well, we hypothesized that they must secrete some developmentally regulated effector that modulate host processes to initiate a productive infection in a new host. So they will also have to inject, right, those effectors to make the host cell a good niche for the infection to actually take hold. And so why do we say developmentally regulated gene? Well, from my studies and multiple other transcriptomic studies, it is clear that Toxoplasma has stage-specific transcriptomes. So there's a sporozoite-specific transcriptome, there's a bradyzoite-specific transcriptome, and there is a tachyzoite-specific transcriptomes, as well as the stages that are in the definitive host. But so from my study, comparing the genes that were differentially regulated between the sporozoite and the tachyzoite during infection, we identified about over 700 genes, and some of them were metabolic enzyme, transcription factor, and non-secreted effectors. But a lot of them were of unknown functions, and among those, there were 85 predicted secreted proteins, and predicted to be secreted because they have a signal peptide, a predicted signal peptide at the end terminus. And I'd like to say that a lot of the genes that we know to be associated with virulence in Toxoplasma were of unknown function, and they were unique to Toxoplasma, as in there's no other organism in the databases that express those genes. And so this was pretty exciting, and I call it the gold pot. So now I have 85 genes that I can go on and play with. So this is just to show you one example of that gene. So this gene is GRA28. When we did the study, we found that in tachyzoite, it is expressed in tachyzoite, but in the sporozoite phase during infection of intestinal cells, the transcript is upregulated 12-fold. And what was interesting is that previously, Peter Bradley's group has found that a tagged version of this protein, GRAD28, localizes to the host nucleus during infection in vitro. And so this is quite exciting. There's another study that basically created a deletion of this GRAD28 and infected animal, but showed no virulence defect. But here's another point about Toxoplasma. There are multiple strains of Toxoplasma. So you have type 1, type 2, type 3, and the exotic strain. And those strains differ in their ability to cause infection in the animal. And so when they did this study, they've done it in the type 1, which is the hyper-virulent form of the parasite. But when we did our study, we looked at it in the type 2, which is the form that is able, at least in the lab, the forms that are able to produce tissue cysts, whereas the type 1 does not produce tissue cysts. So whether or not GRAD28 is important for virulence in the type 2 strain remain to be determined. And we know, I want to add that we know that this, you have genes that are important for virulence in type 1, but not in type 2, but some that are important in type 2 and not in type 1. So there's precedence for this hypothesis. Okay, so to go about studying, identifying effectors that are important for this initial infection, well, there's no way I am doing this at CSU East Bay. There's no cat. As a matter of fact, the US government, the USDA just decided that we will no longer work with cats to study Toxoplasma, and it's too expensive, and the sporozoites are very, 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 very infectious. So I do not trust my undergrads, and so that's not gonna happen. But what is cool is that this stage, the bradyzoite stage, we can get them at CSU East Bay by doing animal infection. We can also extract the bradyzoite mechanically or induce the excitation with pepsin. But what is even more cool or cooler is that in vitro, we can induce the differentiation of the tachyzoite, the rapidly growing form, by starving them. We can switch them to the bradyzoite and get the bradyzoite out. So little caveat, bradyzoite in the tissue cyst from in vivo, they are slightly different from the bradyzoite that occur in vitro. But the in vitro bradyzoite are a good starting point for what we want to do. And then we can validate that in vivo. So this is just to show you an example of a stage conversion assay. So you're looking at human foreskin fibroblasts, HFF, infected with tachyzoites. And the red marker here is a surface protein called SAD1. So you can see that day 1 post-infection, under those conditions, most of the vacuoles, most of the parasites are in the tachyzoite form. But then 4 days later, you start seeing the production of cysts, okay? And how do we know this is a cyst? It's because the parasite are inside, but we've labeled the vacuole with a cyst wall marker, DBA. So anyway, so we can get that in vitro. And then we can also release the parasite from those in vitro cultures and use them to infect intestinal epithelial cells. And so this one is from my lab. We're trying to develop this in vitro system so we have a reliable tool to investigate our questions. So here you're looking at parasites that originate from a switch assay like this, and then infecting intestinal epithelial cells, so IC18. And as you can see, the tachyzoite, you get some tachyzoite infection, whether or not these tachyzoite come from the tachyzoite that were existing in the culture, or whether or not it is a bradyzoite that switched to tachyzoite, we're not quite sure. So we have to we have to improve this assay. But what is clear is that you have bradyzoites that are able to invade intestinal cells and divide as bradyzoite. And we know that they do divide slowly in the brain, but it was not known that they could divide in vitro in intestinal epithelial cells. And so that is interesting. And we also, there are other images that I'm not showing you here that shows that you start seeing the markers of both bradyzoite and tachyzoite on some of those cells. All right, so what are we going to do with all these systems? So the approach we use in the lab, I call it the one gene, one student approach. So we have the wild type tachyzoites, and we can induce them to form the bradyzoite in vitro. We can also use multiple molecular cloning approaches, including CRISPR, to generate knockouts of genes of interest and then perform the same experiment and obtain a mutant bradyzoite, okay? So just this part can provide more than one experiment, right? So first of all, can the mutant switch to bradyzoite? If they don't, hey, good, we have something to study. But if they do, then we can ask the question, how do they behave when you start an infection with the bradyzoite and if you start an oral infection, which is the normal route of infection of the parasite? And so I told you that sporozoite, right? The sporozoite are the form that comes out of the oocyst, and then the bradyzoite are the form that comes out of the tissue cyst. And so I reason that, you know, and this is, please don't judge me too much on this, but if I were a parasite, I don't know why I will reinvent the wheel just because I'm in one form or another, right? So I reason that there may be a shared subset of initiation factors that both the sporozoite and the bradyzoite secrete to start this initiation of infection. And so when we look at the transcriptomic data that is available on ToxoDB, there's a lot of genes that are different between the sporozoite and the tachyzoite, and between the bradyzoite and the tachyzoite. And when you look at the genes that they have in common, we've identified 48 of them that have a putative signal peptide. And again, if the protein has a putative signal peptide, it is more likely to end up in one of those secretory organelles than not. And like I said before, the content of those secretory organelles are really critical for this host-pathogen interaction. And so we've looked at 13 of them, and I'm not going to show you, I'm just gonna show you a brief highlight of the data. So of the 13 that we've disrupted, 9 of them showed no difference in plaque assay, meaning that they can complete the lytic cycle of Toxoplasma. 2 of them had a defect in plaque formation, and then 1 of them had a higher plaque number, okay? Meaning that it grows much faster. And then we introduced them into the animals as in the tachyzoite stage. 3 of them were virulent and 8 of them were virulent. And so the plan is to take those 8 that were virulent and look at them in following oral infection, because it may be that their phenotype happens in the other oral route versus the IP route. Now, here's where my life just started to cry, okay? You see these 2 genes that I'm highlighting over here? When I was at Stanford, they published on this one when I found it. So when I found the phenotype, then a paper came out. Then I came to my lab. First, 2019, they published on this delta granine. So I got scooped twice in this project. So please, people, nobody touches row 23, 28, 70, 40. Anyway, so it's validating, right? Because at least it tells me that the approach that we're using is not that crazy out there. And so what we've been doing is looking at these 2 genes, ROP23 and 287040. So the lytic cycle of the parasite is as follows, right? You take the tachyzoite, infect HFFs, then you allow the parasite to grow, and then eventually they will lyse the cell. And if you do this experiment without moving the flask, you can count the number of plaques, which means the number of parasites that are infected, and you can basically count the number of plaque and the size of the plaque. And so when we compared the wild type tachyzoite to the mutant ROP23 or to the mutant 287040, we didn't see much of a difference in plaque formation. So the tachyzoite form is able to complete the lytic cycle in vitro, okay? Which was as expected, since these genes have higher expression level in the bradyzoite relative to the tachyzoite. So we were really not expecting any phenotype associated with the tachyzoite developmental form. But when we introduced those mutants into the animal and we looked at virulence, we found that relative to the wild type, the delta ROP23 and the delta 2870 mutant were avirulent, okay? And you can compare that to another mutant over here, delta ROP42, which seemed to be more virulent than the wild type, at least in this experiment. And we then took the surviving animals, and you can see there's only one wild type that survived, and we counted the cyst in the brain, and the mutant produced much lower number cysts compared to the wild type. And these are preliminary, as you can see here, N equal 4 mice, so we have to repeat this experiment, but this is promising, and we think that we may have identified two new virulence determinants of Toxoplasma that are under developmental regulation. And so my student Paula, she tried to localize this ROP23, which is supposed to be a ROP3 protein, so localized in the baseball bat looking organelle, and she couldn't do it in type 2 parasite. However, there's a paper that expressed this gene under the tubulin promoter and showed that it localizes to the ROP3s. And so there's we think that there's a regulatory mechanism that is associated with the expression of this gene that matters in terms of that is important in its role during Toxoplasma infection. I think I butchered how I explained it, but yeah. So another student in the lab, Prayasha Roy, she tagged this other gene, 287040, and what she found is that the gene is not expressed in the tachyzoite form in vitro. And in the bradyzoite form, it is at the apical end over here, and which is reminiscent of the micronemes, which are those cigar shape that I mentioned at the beginning of the talk. And the micronemes are involved in invasion, okay, in Toxoplasma invasion into the cell. The other thing that was interesting is that just this year, there's a paper that was published that showed that this 287040 with an HA tag is expressed in the tachyzoite stage of a type 1 parasite. So again, and the type 1 parasite do not produce cysts, and our form over here, the type 2, do produce cysts. And so because we're interested in the bradyzoite form, we think that this TG 287040 may be involved in the bradyzoite invasion of the host cell. And so trying to reconcile this expression pattern to this virulence result would be quite interesting. And I can speculate what I think is going on during the Q&A. So this is an example of how we are approaching our goal to identify stage-specific effector protein. Now, I'm going to tell you a little bit about some of the work we've done trying to look at differentiation between tachyzoite and bradyzoite. So I put this little cartoon over here, and I didn't talk about it previously, but sporozoite will convert to tachyzoite without any division. We don't understand any of this process. Tachyzoite will convert to bradyzoite, and we do understand some of the regulation that is involved in this process. So the tachyzoite to bradyzoite conversion is under transcriptional regulation by factor like BFD1 or AP2, Apical 2 type transcription factors. And so we know a lot about this, but how bradyzoite goes back to tachyzoite, which is what needs to happen when the parasite initiates infection, we don't quite understand it, okay? And so these are the two paths that we're looking at: sporozoite to tachyzoite and bradyzoite to back to tachyzoite. And so if you look at those EM again, you notice these starch granules, right? So these starch granules are full of amylopectin, and amylopectin is a starch. You find it in French fries and potatoes, and it's in human, it's considered to be a source of energy. And so you have accumulation of starch granules in the sporozoite and in the bradyzoite, and you can barely see it. You can barely see them in the tachyzoite. Now the question is, how does the parasite goes from all this amylopectin to no amylopectin within 12 hours, right? Because in vivo, it was shown that the parasite, sporozoite infected mice, the parasite will go from sporozoite to tachyzoite within 12 hours. So this process is fast, and we don't know. We don't know how these amylopectin get formed. We don't know how they're regulated. We don't know how they get used. And so this is what my previous grad student did her master's on. So a lot of these glycolytic enzymes, so enzymes that are involved in amylopectin biogenesis, are present in Toxoplasma. And a lot of them are under transcriptional regulation. So here we basically looked at the data from Pittman et al. From Loran's lab, where they infected animal and look at the parasite during acute infection and during chronic infection. And as you can see, the hallmark of bradyzoite that we know, so NLS1, LDH2, that are upregulated. And then you have this protein, GPM1, that has its isoform GPM2, and then glucose phosphorylase, GP, that are also upregulated in the chronic phase. So we know from Sugi et al. That glucose phosphorylase is required for efficient stage conversion. But we don't know anything about this. And when you look again at the transcriptome of Toxoplasma, there's over 189 genes that are associated with starch, sucrose, maltose, glucose, carbohydrate, essentially, carbohydrate metabolism, that are under developmental regulation. So what do they do? Emily, the student, decided to look specifically at GPM1 and GPM2 because it is at the interface of glycolysis and amylopectin production. So GPM1, glucose phosphate mutase, catalyzes the conversion of glucose 6-phosphate to glucose 1-phosphate, and it's reversible. And so glucose 1-phosphate can be converted to glucose 6-phosphate and then go through glycolysis, or glucose 6-phosphate can be converted back to glucose 1-phosphate to enter the amylopectin biosynthesis pathway. And here you see that expression of GPM1 is significantly upregulated during the chronic phase of infection. And so what Emily did, she created a knockout of GPM1, and she found no difference in plaque, in the formation of plaque in the tachyzoite. I mean, there's a decrease here, but it wasn't statistically significant. And then we measured the plaque sizes, and we found no differences as well. Then we performed switch assay, converting the tachyzoite to the bradyzoite, and the mutant were able to switch just fine after 4 days. And preliminary data looking at the amylopectin staining, which is what this periodic acid shift allows us to do, we see that the mutant is able to accumulate the amylopectin similar to the wild type. I mean, we still have to quantify this, but at this point, it doesn't seem like there's much differences. The only slight difference we saw is that there was a difference in the cyst size, but I think it's a numbers game here, and we have to do a bit more more experiment to confirm this, either taking the experiment longer or just, yeah, I guess that's my take on this. So the other thing we've done, because GPM1 is associated with glucose, if you will, we decided to say, okay, well, maybe we've performed because we performed these experiments in the presence of glucose, maybe if we remove glucose from the media, we will be able to see the contribution of GPM1 to Toxoplasma biology. And so there's an assay called a replication assay, where you infect the human cells with the parasite, and you wait couple hours, and then you count the number of parasite inside the vacuole. So this vacuole has one parasite. This one is basically going to produce 2 parasites, so 2 vacuole, and then this one is going to have 4 parasites. Okay, so this is 1 division, 3, and so that's 3. So here you have a 1-pack, a 2-pack, and this is a 4-pack. Okay. So this is what we've done here. And so when we count when we infected the cell for 24 hours and we count, this is the wild type and this is the mutant, you can see that there is a lot more APAP in the mutant compared to the wild type, both in the presence and absence of glucose. As a matter of fact, in the absence of glucose, it seems like the mutant is growing much faster. So this is preliminary, and personally, we're still scratching our head to try to think about what it means. But it can be that in the absence of GPM1, there may be more nutrients available that are not diverted to storage of amylopectin. But again, there's a lot that we can sort of dissect based on this experiment. And so I think I will stop here. And so I've shown you that Toxo employs some stage-specific protein to interact with the host, and there's a lot that we don't know about those initial infection. I've showed you that this approach, looking at a transcriptionally regulated gene, can allow us to identify novel virulence determinant of Toxoplasma. Then we can also look at developmentally regulated metabolic enzyme like GPM1 and uncover new features or new biology associated with this parasite. And clearly we have all the tools that, at least in my lab, that we can use to answer those questions. And so I'd like to acknowledge my amazing students. A lot of this work has been done with them. Paula over here did the Rope 23 study. Priyasha did the 2870-40 study, and Address, Ariel, and what is his name? Zach, working on the metabolic aspect of it. But anyway, a lot of them have their own little story that fits in the grand scheme of the lab. And I want to highlight Bin Kao over here. He was one of my first undergrad, and he's now at UCSF Medical School. So that make me represent the Guiton lab at UCSF. So that's, yeah, and my collaborators and funding sources. Thank you for listening, and I will take any questions.
Great. Nice job. Thank you, Pascale. Excellent. So we have just a few minutes to take some questions from the audience. I just did want to leave a little bit of time for Pascale to talk about a group that she co-founded this summer called the Alliance for the Black Community. But are there any questions that we can take from the audience at all?
So there's one here that I can answer.
Okay.
Would GPM2 compensate for the lack of GPM1? Is it possible to generate a GPM1 to DO knockout? Yes, absolutely. So this is something that we're working on, and Emily she created the construct for the GPM2, and we are going to make the double knockout. And we know that a GPM1, GPM2 double knockout can be made, at least in type 1 and type 2, because our collaborators in Germany, they've made that, but they didn't see anything in the tachyzoites. So but they didn't look at different conditions. So there's room to play there. But we think that GPM1 may be compensating for the absence of GPM2. Thank you, Constance. How do you think toxoprotein expression may change in an intestine with the microbiome co-infection? That is a good point. I know that depending on the mouse strain, the virulence of the parasite is different. And we know that the animals have different microbiota, and so there's definitely some toxo-microbiota host interaction. But how it will change, I don't necessarily know how, but I pretty much know it will change for sure, right? Because it's going to take cues from the microbiota, and yeah.
Great. Robin also has a question. She can ask live if she likes.
Hi, great talk. I was just wondering if you had looked at any of your effector genes, how they affect the host, or do they affect the host in any?
Yeah, so that's why we are going. So ROP23, the idea is that ROP23 is a ROP3 protein, and ROP3 protein are injected directly into the host cell, and then they will go and basically modulate the host. And so what we're trying to establish is that some of these protein, we're trying to establish which protein are secreted outside the parasite. And then when we establish that, we can basically overexpress the protein in the host cell and see if there's any change in the host, any changes in the host transcriptome. Alternatively, we're planning on doing, on taking those knockouts and repeat the transcriptomic study, looking at the effect of deletion of those genes on the host transcriptome compared to the wild type. And so this is how we're gonna start addressing whether or not these proteins are exerting the effect on the host or doing it in the parasite. So have you looked at, oh yeah, that's her question. Does that answer your question, Robin?
Yeah, no, that's really great, really cool. Curious if you're also gonna look at any signaling changes, not just at the transcriptome level, but
protein.
At the protein level.
Yeah. So that's in terms of proteomics. At this point, I do not have plan to do proteomic, but we're gonna look at basically if we identify a particular gene that is giving us a strong phenotype, like the ROP23, for instance, we are going to study that at the protein level as well, but just one protein at a time.
Yeah, really cool.
Great. I think Cynthia Martinez has a question for you.
Yeah. Do the amount of starch granules differ between different forms of toxo? Do we know if the content differ between these forms? Yes. So we know for a fact that the starch granules in the sporozoite and the bradyzoite are way more than the tachyzoite. The tachyzoite you can find 1 to 2, but in the sporozoite and bradyzoite individual cell you can find up to 10, or even I think Duby, when they did this study, they actually counted the amylopectin granules here, and it ranged from, I don't know, 5 to 20, and if not more. But yeah, so there's definitely much more. In terms of content, so far all the reports that I've read, mainly amylopectin.
Okay. I have a question for you, Pascale. You know, like you said in the beginning, Toxo is just really famous for a lot of things, just like the Kardashians, except it's going to last longer than the Kardashians. But I was wondering, you know, Toxo is known to change host behaviors, and is there do they know why it has a predilection for the brain cells?
So I don't know that I know why, but the evolutionary argument is that the purpose of life, right, is to pass on your genes and to maintain your species. And so the only way Toxo can get to its sexual cycle to ensure diversity in the gene pool is by getting into the cat. So the idea, the evolutionary argument is the parasite gets to the brain so it can essentially manipulate the host to get back into the feline, right? And so the evidence, at least in the rodent, it's pretty strong that that happens. So Vyas, I think it's Vyas group, they've infected mice with Toxoplasma, and then uninfected mice, and then they exposed them in a quadrant to rabbit urine and to bobcat urine and then home. And so what they found is that the cats that are chronically infected with Toxoplasma, that is, they have Toxoplasma in their brain, will tend to spend more time in the cat urine quadrant. So they spend more time there. And the argument at the time was, oh, they lost the fear of the cat urine, or they are attracted to it. So I'm not a neuroscientist, but what I gather is that the pathway for attractiveness versus fear versus aversion are different. And so I don't quite know which path it is, but for one thing is sure, they tend to get closer to the cat, so more likely to get eaten.
Great.
So yeah.
We have just a few more minutes here, and I would like you to speak to the group that you co-founded, which is the Alliance for the Black Community, or ABC group, in which you've developed some educational documents. So would you like to talk to us a little bit about how that started and
Sure.
what you're doing?
Sure. So I am Black and I am proud, and I want to start by saying that there is the scholar strike right now for racial justice, and my way of striking is by talking about racial justice in my class this afternoon. And so anyway, after the death of George Floyd, actually for me it started with Ahmaud Arbery. I've been very I've been affected by this. I want to share my screen because I get some cues. I've been quite affected by it, right? Mentally, emotionally, just like most people. And then, to be quite honest, I may get in trouble for this, but the letter that our president committee sent to the university was very disappointing to me. I absolutely hated it. I felt like it was just, again, pandering, and because there was no word for the Black student. And so with my colleague, Nazi Papour, we wrote a letter that was addressed to our Black student at CSU East Bay. And we had over 600 faculty sign the letter, and then we sent it to the student. And as a result of that, Nazi and I decided that, well, we cannot just sit there and keep signing letters. We have to start doing something. And this is how the Alliance for the Black Community was born. And it's all volunteer. We don't really do, it's a bunch of faculty and staff from CSU East Bay that are trying to make things better for our Black community, both faculty, staff, and student. And so some of the work we're doing is to try to demilitarize campus police. We're trying to improve the education of our Black students. And the reason is at East Bay, the Black student population is the population that is essentially not succeeding. And so we have to do something about it. And also in the education portion, we're trying to develop material to help faculty develop racially sensitive curriculum that actually includes students, right? And so as part of that, this summer with my students in my lab, Alpana is a high school student and Ariel's an undergrad, we put together this atlas of Black scholarship for inclusive and racially diverse STEM curricula. So this document, it allows you to find Black scholars that are in a particular field, immunology, microbiology, chemistry, biochemistry, and use it in your classroom. Not to say, oh, this is the first Black person, but to say, this scientist did X. So I'm really trying to normalize the scholarship of Black scientists, just like that of white scientists like Pasteur and Robert Koch and so on. And then of course, we focus on Black faculty hiring, retention, tenure, promotion. And this one goes straight to my heart because I have faced discrimination and racism. I was told to go back to Africa in one of my student evaluation, which is, and so I definitely feel like there's a lot that needs to be done. And so we just created this document, Anti-Racist Educator Starting Guide, to basically try to have department start talking about racism in the department and having clear 10 points of action that they can do to make, to have an anti-racist department essentially. And so with this, nobody can say, I don't know how to start talking about racism. You have a guide, so you can do that. And then I also started this Black indigenous people of color group in STEM for our student in STEM, so that we can come together and talk about what it means to be Black and indigenous and a student of color in STEM. I know from my experience, it is isolating and you need good mentor. And yeah, I wouldn't be where I am today without the likes of Zehava and John Bufroyd and Scott Holgren, Capron. So I just felt like my student need an environment to be Black and to be student of color and be unapologetic about it. I'm just tired of always walking and trying to justify my existence in academia. And it's just, yeah, anyway, I can go on and on about this, but that's that. That's what we do.
Thank you very much, Pascale, for all you do. It is at the end of the hour, so let's just say thank you so much for a great talk and what you're doing.
Thank you, thank you. Thank you all for coming. And yeah, you can find my email address on the CSU East Bay website, and yeah.
Great. Okay, thank you, Pascale.
Thank you.
Thank you everyone for joining.
Yeah.